Okay, let's get started. A couple of quick announcements before we get any further. We'll be talking a little bit about the double helix today. Maybe you can't see Jim Watson up at the top here. How many of you read this book? How many of you know how ridiculously egotistical it is? One of my colleagues claims that Jim Watson was hitting on her at one point in the Cold Spring Harbor bar, which, since she was married at the time, kind of gives you a bit of an indication of the particular person here. Second thing is in the middle. I'm sure all of you read Slate and go down, scroll all the way down to the science section. This was posted, I think, 2 days ago. I just published, together with a couple of colleagues, a review article on astrovirology. And this is just one of the press things that's come out on it. I don't like they should be starting with viruses really, but much more importantly, these not quite life forms have a bad rap. As far as I'm concerned, viruses do have a bad rap. So happy to talk about that offline, and I'll post this as well, excuse me, on the class D2L site. So last time we talked about the Hershey-Chase experiment, right? Yes? What was that? And we're all feeling happy about it, which means he's about to ask you a clicker question about it, which you all saw last time, but I deleted out of the lecture notes if anyone went back to look for it. So you may or may not have noticed that. Oh, and that also one other quick thing. I don't know how many people are going and looking at the lecture notes on YouTube. Is that useful? Yes. The second question I have about that has to do with this pointer thing that shows up on there. The reason I'm using my phone is it means the pointer ends up on those recorded lectures. If I were just using a laser pointer, it wouldn't show up. Is that useful? Yes. Okay, good. So we'll deal with technical issues as we come through, because otherwise the pointer's not going to show up on those recorded lecture notes. So I will keep trying to use this pointer thing. Okay, so that being said, clickers, I know a number of people have asked me questions about clickers as well. Online, things are not getting recorded, etc. The problem, to some extent, is D2L. If you have 0 points, it still gives you a dash as if you have never registered your clicker or gotten anything. So everybody who's here and uses their clicker today is going to get a point for just participating. Yay, woohoo. But that way you can also check and make sure your clicker is working. If it is not, even after today, and I've uploaded the scores, send me an email with your username and your clicker number, and I will make sure that you are appropriately registered. So with that, we'll do our first clicker question, assuming that we can get this to actually move. Yes, good. So if Chase and Hershey, because it was Martha who really did the experiments, had found that S35 was passed from old virus to new viruses in their experiment, they have concluded that DNA was the genetic material, RNA was the genetic material, protein was the genetic material, they had contamination, or the viruses contain protein. Again, feel free to discuss this. Sulfur was passed. S35 was passed. Given the experiment that they did, or experiments, I should say, that they did. Okay, what do people think? Do I agree or not? Let's check. So what do we have? 85%. We're getting close. 100% is what I'm aiming for here. And if I find out who's screwing up my 100% sometime, I'll come and find you. No, just joking, really. It closes automatically in a minute, and it counts down. It was timing as we had that whole minute there. It's right next to this piece up the top here. Oh, yeah, right there. You can't see the multiple choice? Okay, well, I'll just show you what it looks like. We'll do another one here. So there it is, 59, 58, etc. So this one is not going to count. So Are we going to try to go for 100% on here? Okay, so that's where that countdown will be. And sorry, I didn't usually see the 10, 5, and 1. I didn't do so this time. Who's messing with me? 2 people are messing with me. 3. Okay, yes, I know. So I will quickly hide this so that you can't see it anymore, and we can stop. Okay, so I will keep the numbers from the first round of voting because I will still have those saved up here. Okay, so now, again, Watson and Crick. A lot of this is what Jim Watson said he did, which may or may not be completely true. Watson is the one on the right here, the crazy American in England at the time. Interestingly enough, his research was funded by polio research. So don't tell any of the granting agencies that when you say you're going to work on polio, you actually end up working on coming up with models for the DNA structure. So I don't think they're complaining that that's what happened, but it's maybe not exactly what they were expecting here. So let's talk a little bit about what they knew. This is all of the background information that Watson and Crick had before they came up with their model. They knew about the structure of the nucleotide, which we'll look at on the next slide here in just a second, and that's actually rather important in terms of thinking about 5', 3', molecular biology, polarity, etc. Particularly this whole idea that the linkages in DNA were from 5' to 3'. And so, again, we'll talk about that structure in just a second. That linkage was known to be a phosphodiester bond, and they'd heard Edwin Chargaff come and visit, and again, it's in the book, is described that Chargaff kind of blew them off when he came and visited Cambridge at the time, because these guys didn't really have a clue. The people who actually really did have a clue were mostly Rosalind Franklin, who was studying the structure of DNA. And really, it was Rosalind Franklin who came up with a lot of the techniques, the mathematics, etc., to really make the breakthrough. And in the book, and actually, as all of you have read the paper, of course, at the end it says, mostly based on published work. Well, the part that it wasn't actually based on was this work here, which was Rosalind Franklin's. This is the really famous picture here of the double helix, the B form. This is actually the A form structure of DNA that we'll take a look at in just a second here. This is not your classic X-ray crystallography structure. A lot of people say this is an X-ray crystal structure of DNA. No, it's not an X-ray crystal structure of DNA. The X-ray crystal structure of DNA came along later and said Watson and Crick were wrong, at least in their general structure. What this is literally a fiber of DNA, and we talked a little bit about last time that one of the things that makes DNA very different is it's a long molecule, a very large molecule, and it's very different in terms of its dimensions. It's long in one dimension and very short in the other dimension. And this was already known, and so basically what Rosalind Franklin and some of their coworkers had worked out is you could basically stretch this out. If you could stretch it out, then you could align it in an electron beam and then look at the diffraction. And so the diffraction here, and we'll look at this again on the next slide in just a second here, gave the information that it was about 2 nanometers in diameter. And there were major repeat structures at both 0.34 and 3.4 nanometers. And this is the data, again, from Rosalind Franklin that they may or may not have stolen the look at, and Wilkins may have showed it to them. Again, a lot of that's lost in the depths of history. The other thing that they had was our favorite son, Linus Pauling, who had already figured out all these hydrogen bonds, beta pleated sheets, alpha helices, etc., and he'd come up with a model for DNA which turned out to be totally, utterly, and completely wrong. He had the phosphates in the middle and the bases sticking out, which logically actually makes a lot of sense, because the bases are the genetic information. You need to be able to access them. So having the bases exposed actually makes a lot of sense. And these upstart American and his British ex-physicist colleague really didn't have a leg to stand on, and they weren't the big guy on campus as far as understanding lots of molecules as opposed to Linus Pauling. So these are just sort of review what they knew. The important thing here is really the deoxyribose, which is present in the middle. That was Arnold Levine. Actually, in 1929, he showed this, so almost 30 years before they came up with their structure. This 2' deoxyribose, there are 5 carbons in deoxyribose, labeled as 1', 2', 3', 4', and 5'. Why are they all named prime? It's because the nucleobase is where you have the original numbers. So you number around here, depending on if it's a purine, which is the big ones. That goes 1 through 7 in terms of the main atoms. Pyrimidines, 1 through 6. So 1 through 9 here, 7, 8, 9. And we'll talk a little bit about where those are a little bit later on. I don't expect you to remember these numbers. I can't remember all of these numbers for the bases. However, for the ribose, I do expect you to remember them. So the 1' position is where the base is attached to the deoxyribose. This is also called the glycosidic bond right here, where we have base attached to the ribose. 2' position, it's 2' deoxyribose. In RNA, you have an OH here instead of an H, so deoxy. 3', this is where you're going to get extension of your molecule. 5' is where the phosphates are attached. So we talked about ATP last time. ATP is phosphates attached at the 5' position. And TP is 3, DP is 2, MP is just one of those. So this is what they knew. They also knew the structure of the purines and pyrimidines. What else did they have? Rosalind Franklin's data. This is the B form part of DNA. And a couple of things that you can see from that structure. I should say Rosalind Franklin could see. I wouldn't have a clue if I looked at an electron diffraction picture like this. Although, to Watson and Crick's credit, Crick in particular was thinking about X-ray diffraction from helical structures. And so he actually had a really good handle on what an X-ray diffraction pattern should look like from a helical structure. So he can sort of take one look at this and go, oh, this is what it really means. And that's what basically is able to see here, that you have these dots here in an X pattern means that it's helical. That you have a big spot out here, that's that 0.34 nanometer repeat. And then these individual spots in here, the closer and closer they get to the middle, this is a diffraction pattern. So short distances means actually long distances apart. Long distance apart in your diffraction pattern actually really means relatively short distances apart. So again, this is the repeat every 0.34, and each of these are every 3.4 nanometers. And this is how they ended up, again, coming up with their structure here. I didn't bring my copy of the biography of Rosalind Franklin. Way better book than the double helix. Highly recommended as far as that's concerned. So again, we're still having technology issues here. So the connection here, back, not forward. There we are again. 5' to 3', just in the individual nucleotide. All of these are connected to each other in the same way. So if you have a polynucleotide chain, the 5' end is up here where you have a phosphate. Here's your nucleotide with the base at the 1' position attached through 3' to 5' to 3' to 5' to 3', etc. So anytime you have a polynucleotide, i.e. more than one of these nucleotides attached together, you're always going to have a 5' end and a 3' end. And again, so the individual nucleotide, they're always attached in the same way. They're always going to be like this. When I say always, somebody should hold their hand and say, Dr. Stedman, but this is biology. There are always exceptions. Yes, there are exceptions. We'll talk about them later on. So what did they do? Anybody recognize these guys here? Monty Python. Can you read what's down here? It's only a model. So this is literally what Watson and Crick did. They built a model. And this is a picture of that model. The model itself is about this big. There's a copy in the Smithsonian. Definitely worth taking a look at. Really pretty amazing. They were also very fortunate that they were in Cambridge, in the UK, where they had a machine shop that would literally make each of these individual pieces. And then they had some clamps, and they were just literally putting them all together. And this was the model that worked. It fit all of the data, and that's where they came up with their famous double helical model. They discovered the structure of DNA. I think that's even what's in the textbook. No, they came up with a model that fit the data. It's not really that they discovered the structure of DNA. They had a model that fit the data. That model has held up extremely well since then. But there are a number of issues with that. I don't know how easy it is to see here, but if we take a quick look right here in the middle, look at this. Oh, hang on. Back up one more. So this is the problem with this pointer here. Here's a G and there's a C. They're the hydrogen bonds between them. Here's the T and the A, the two hydrogen bonds between them. Do you see something here that looks a little weird? In terms of what you know about the double helix? No, yes, confusing. Look really carefully here at this GC base pair. What do you see here? There's a hydrogen bond, there's a hydrogen bond. There's not a hydrogen bond. And this was apparently Crick's idea, is if you're going to have something that's so beautiful, you can't have stuff that's got different numbers of hydrogen bonds between the bases. That's not going to work at all. So everything is nice and symmetric. Has to be nice and symmetric. So this is what they came up with. This is again from the paper. Again, from their model building, the red ones are the important things, complementary strands. And so as far as I'm concerned, this is really the big breakthrough that they made through their model building. And that is that there's no way from Rosalind Franklin's data that you could tell how the strands were oriented relative to each other and how they were relatively speaking. And so the idea of complementarity, that if you have one base on one strand, it has to give you the other base on the other strand. So if you've got a T, it's got to be an A. If you've got a C, it's got to be a G. That's the only way it fits into the structure. So complementarity was really something that they came up with, again, but putting their pieces together and see if they would actually work. Anti-parallel, when we say anti-parallel here, we mean that if one strand is from 5' to 3', the other strand is going to be 5' to 3', the other one opposite to it will be 3' to 5'. They're always going to be opposite and anti-parallel to each other. This is the whole point of the molecular biology polarity. So if you're one direction on one strand, you're going to have the opposite direction on the other strand. And so I've got my model over here. As that I've mentioned a number of times, it's got some problems with it. Come and take a look at it afterwards if you want to take more details on that. Phosphates on the outside, that was kind of a guess. They'd seen in Linus Pauling's structure that the phosphates were on the inside. Here they said they were on the outside. It seemed to make more sense. But then immediately you have the problem of how you access the information. If the information is just in the A's, T's, C's, and G's, then how do you access that information in the middle? And these hydrogen bonds are perpendicular to the backbone. And so we can just take a quick look here. Here we've got our model, anti-parallel strands, so going like this, 5', let's see, here's a 5' up there, 3' down there, and anti-parallel. You have phosphates on the outside. These are the yellow ones, and again, people can come up and take a look at this later. It's also falling apart. Hydrolysis is happening here, as I can tell, as we're watching. And the bases are perpendicular to the phosphodiester backbone on the outside, literally right in the middle here. There are a couple things they had to do, and they mention this also in the paper. C4 of cytosine and C6 of adenine are amino, and C6 of guanine and C4 of thymine are ketos. And that's, again, those numbers I can't remember, so I don't expect you to remember them either. But basically, amino groups rather than imino groups, so NH2 rather than NH, and double bond O and not OH for those. Because of their structure, and again, this is something that they came up with from their model, something you couldn't have gotten from the X-ray diffraction data, are major and minor grooves. And so here, a big gap, this is a major groove, minor groove is over here on the other side. And we'll take a look at this in more detail in a couple more slides as we keep going on here. It's also a right-handed helix, in fact, right-handed double helix, both of them wrapped around each other. This is a nice model because I like to call it the right-armed helix, because I can stick my arm in the major groove. It goes up. If I try and do the same thing here with my left arm, it doesn't work. And I have a whole collection of various different helices over here on the side table if you'd like to come take a look at them later. If you're a little confused about right-handed versus left-handed helices, there's a number of left-handed helices over here as well. We've got this room for an hour afterwards, so office hours will be here, so you can come take a look at them afterwards. You'll have a little bit more time. On the side here is just shown this, that we have amino, not imino, and keto versus enol. And that's the only way you can make this work, in such a way that the base pairs are all the same inside this double helical structure. So what does the base pair structure look like? Again, G's pair with C's, A's pair with T's, and the distance here between where these sugars are. Remember I told you about the glycosidic bonds? Here's one backbone, here's the other backbone. This distance and these angles are basically identical to each other, whether you have an AT or a TA, a GC, or a CG. So the scaffold is your phosphodiester backbone, the two helices, and you can basically put whatever information in the middle you like to have there. Is everybody happy on base pairs, DNA structure? There will be a question about DNA structure on the exam. Probably if any of you have looked at the old ones, you'll notice that there have been questions about DNA structure on the exam. Years ago, I used to ask everybody to draw a base pair for me. I got a little fed up with grading 150 base pairs, and most people ended up missing things because even these images here, these two particular base pairs have problems. They're missing a couple of hydrogen atoms, etc. So it's hard to find them that actually have exactly the correct kind of base pairs. So take a look. Think about what's missing on all of these things and what's correct. So any more questions on this? And we'll try the, and I'll try to give you the countdown as well on our next clicker question. So Watson and Crick's original model for B-form DNA had how many hydrogen bonds between guanine and cytosine? 2, 3, 4, 8, or 10. This should hopefully be very straightforward. 100%, except for that whoever it is who's messing up my statistics. Ten, 5, 'kay, nobody liked 8 or 10. Oh, go figure. I'm not quite sure we get 4 hydrogen bonds. That would be interesting if we're talking about A's and T's and G's and C's, if we're talking about those. But the original structure, I should say the model which they came up with, did actually have just 2 hydrogen bonds between the G and C. And the reason for that was probably just because it was prettier that way. And we now know this actually, again, not to be the case, but that's what they liked and they showed their structure originally for. Also shows that they weren't really chemists, unlike Linus Pauling, which I think was one of the first people to point out that they were incorrect about this. So to risk beating the dead horse, as it were, here, polarity of individual nucleotides, of strands, always 5' to 3', and always anti-parallel if you're looking at the two strands here. So if you've got a 3' end of one of the strands here, the opposite one is going to be 5'. 5' here, 3' here. And this is just drawn out here linearly. Again, 5' here, 3' there. And the main reason for this is that it gets these glycosidic bonds into the same place so that you can have the base pairing take place. If you were to flip it over and try and do it the other way around, they're not going to fit. It's going to be a really messy structure. You're not going to have the nice continuous double helix which is on either side there. Of course, everyone shows these nice, pretty pictures, models of something that looks like this. What DNA really looks like if you were a protein and appropriate that particular scale. This is what DNA is mostly going to look like. Of course, this is a static view. It's moving around because of Brownian motion, all we're in aqueous solution. But it's pretty full. All of the reds here are oxygens, which has what kind of charge on it here? Because it's DNA. These are all going to have negative charges. And then all of the base pair information is here. All the blues are nitrogens. These are all present in the individual bases in the middle of the DNA, to some extent protected from the rest of the world, as it were, due to these phosphates on the outside. So all the information is here in the middle of your DNA. Okay. So just another way of looking at this, major groove, minor groove. This is actually zoomed out a little bit here. If you look at again, the structure for the model has one large area and one small area. Why do we care about this? It really has to do with the fact that all of the interesting parts of the base pairs, i.e. all of these differences in terms of chemical groups, are going to be sitting here in the major groove of DNA. And we'll take a look at this again on the next slide. But here, if you look at thymidine, methyl group present on thymidine. If you're ever trying to find a major groove on a piece of DNA in B form, look for methyl groups. I'll give you a quick hint. It's a big black dot on this structure right here. So if you're looking for major and minor grooves, that's a really good way to do that. And the only place you're going to find a methyl group in DNA is going to be in the major groove. So what that tells you is the specificity for what kinds of bases you can see on DNA. When see, I mean proteins coming in from the outside. That's going to be things that are present in the major groove. I mentioned, right, when we talked about Rosalind Franklin's data. Oh, and one other fun fact about Rosalind Franklin that's important to me and not to you, so it won't be on the exam. She went to the same college as my mother did. So, yeah, special question for the exam. No, I promise you that question will not be on the exam. So, anyone asks me what's not going to be on the exam, that won't be on the exam. But yeah, so when they were looking at these crystal diffraction, I say fiber diffraction, I say they're not crystallized. Fiber diffraction, they had two kinds of DNA, and in fact the best data they had was actually from A-form DNA rather than B-form DNA. We now know what the structure of A-form DNA is. It's actually also double-stranded, also wrapped in right-handed double helices, but instead of the bases being nice and perpendicular relative to the backbone, they're really squished and sideways. Now, why should anybody care about A-form DNA? It's just what Rosalind Franklin showed. Well, as I mentioned, this was a model that Watson and Crick came up with for that nice data for the B-form DNA. Turns out DNA in cells is all kinds of different forms of DNA. It's a mixture of B-form DNA, A-form DNA. There's also Z-DNA, which has a left-handed helix on it. And A-form DNA is also present in the genomes of some viruses. So some viruses actually package their DNA in this form rather than this form. In fact, one of my colleagues published a really nice paper showing that that was the case here. But you'll notice it's really much more compacted. It's a more compacted form of DNA, which, particularly if you think about a virus, usually it's pretty small. It's nice to have a relatively small structure to work with there. So as I mentioned before, major groove, minor groove. If you look at a double helical piece of DNA, just the different base pairs, CG, GC, TA, and AT. All of the interesting stuff is in the major groove. We've got hydrogen bond acceptors. We've got hydrogen bond donors. We've got methyl groups. And what that means is that if you have something that needs to interact with DNA specifically, i.e. in a sequence-specific manner, you interact with the major groove because that's where the chemical information is that you can interact with. Now, what do you think might want to interact with a major groove of DNA? Pardon? RNA is a possibility. Doesn't seem to be as much. What else? Polymerase, which is a subset of what? Proteins. Turns out, and this is probably not a random turning out here, but the process of 4-odd billion years of evolution, an alpha helix fits perfectly into the major groove of DNA. Where are the side chains on alpha helices? Sticking out. So they are going to be able to interact with these hydrogen bond donors and acceptors, methyl groups, etc., which are hanging out in the major groove of DNA. So just another way of looking at this. Here, people talk about this, the DNA recognition code. You have hydrogen bond donors, hydrogen bond acceptors. No, I don't expect you to remember this absolute detail. That, let's see, a GC has a hydrogen bond acceptor followed by two hydrogen bond acceptors, hydrogen bond donor, and a hydrogen atom. I would expect you to remember that there's a methyl group sitting in the major groove of DNA. And if you just look again, look for major, if you're worried about major and minor grooves, you want to come take a look at them, come take a look at the model here right after class. So people clear on major and minor grooves? Yes. We ready to ask a question? We've got our clickers out. Good. Hopefully it's not too much of a giveaway when I'm going to do these things. Most sequence-specific interactions between proteins and DNA disrupt base pair hydrogen bonds, are to the backbone phosphates, or to the backbone ribose, are to base pairs in the major groove, base pairs in the minor groove. The other reason to do clicker questions is allow me to have a drink. Ten, 5, not quite 100%. We're working on it. We're definitely working on it here. So what do we have? 97%. Again, someday we'll get 100%. But what I should do is actually stop the scoring. I've got a little score keeper up here at the front, so it gets 100%. I'll just stop it. So I make myself feel better about it. So yes, it is base pairs in the major groove. We will see, and one of the things to note here, I said most sequence-specific interactions between proteins and DNA are to base pairs in the major groove. There are actually some that interact with the minor groove, but it's only in combination with things that are interacting in the major groove as well. And we'll talk about those later on when we get to those particular examples here. So I want to talk about a little bit about some of the aspects of DNA which turn out to be incredibly useful for, oh, sorry, question? Yeah. Yes, the methyl group is sitting on the thymine. Yep. That is the only one that you will see in base pairs. And it's a good review if you're thinking about, no, in terms of thinking about structures, yes, I'm not going to ask you to draw a base pair on the exam, but it's a really good thing to practice. Just that I can't get around to grading all of them. So there are a couple of things about DNA that makes it incredibly useful from a just experimental point of view. And one of those actually really depends on the structure of DNA and spectroscopic principles of DNA. Who cares about spectroscopic principles of DNA? But this tells you, in fact, a lot about how the DNA is interacting, but also allows you to see if you've got single-stranded or double-stranded DNA. The DNAs are together or they've come apart. And the reason that that's so interesting is I'm going to take my model here, so it's maybe a little bit hard to see, bring it out to the middle. If you look at double-stranded DNA, what do you think most of the stability comes from? Pardon? Hydrogen bonds? Covalent interactions on the backbone. Actually, it turns out most of the stability in DNA comes from stacking all of the base pairs together. If you look at the structure of each of those purines and pyrimidines, there are lots of ring structures. Ring structures love to pack on top of each other. And so most of the stability that you have in DNA actually has to do with stacking of the individual base pairs. And because of that, it means that they're sharing electrons. It changes the absorbance at particularly relatively low wavelengths. So if you look at double-stranded DNA, oops, back up here, it's got a relatively low absorbance at 260 nanometers relative to single-stranded DNA. But even in single-stranded DNA, you have this base stacking that takes place. So if you're just looking at a single base, the molar absorbance at 260 nanometers, this is 1.6. I don't expect you to remember these numbers. I do expect you to remember that double-stranded DNA is lower than single-stranded DNA. This is also an incredibly useful tool if you're in the lab. If you have an A260 of 1, it's 50 micrograms per mil of your double-stranded DNA. So if you're taking a lab like the Advanced Molecular and Cell Biology Research Laboratory next term, which is one I'll be teaching, we also call it mutant viruses from hell, then this is an incredibly useful number to know, the 50 micrograms per mil of optical density at 0.1. But that's not why I wanted to talk about that. What I really wanted to talk about is having to do with pulling strands apart. As I mentioned before, one of the really nice things about Linus Pauling's incorrect structure is the base pairs were all pointing out. So it was really easy to have sequence-specific interactions with them because you could form all the base pairs there. But if you're just forming interactions with base pairs that are double-stranded, then that becomes a little difficult. And particularly if you're thinking about replicating DNA, what do you need to do? You need to pull the two strands apart. And it turns out that because of this base stacking interaction, which is an incredibly energetically efficient way of putting DNA together, if you have a bit of single-stranded DNA, it turns out to really destabilize your whole double-stranded DNA. So you have a transition between double-stranded DNA and single-stranded DNA, which is really very rapid. And one way of looking at that is here. If you just take a DNA solution, has a relatively low value at 260 nanometers. What does that mean? It's double-stranded because that's where you've got the lower absorbance. If you heat this DNA up to a certain temperature, these two strands come apart, and you can see that if you just follow the black line, that is your absorbance. And the fact that this goes very quickly from double-stranded DNA to single-stranded DNA says that as soon as you start to pull these apart, they'll come apart very rapidly. The other thing that you can see with different experiments along these lines is that where the halfway position is here, the so-called melting temperature, or TM of your DNA, this is completely dependent on the relative amount of G's and C's and A's and T's that you have in your DNA. So if you have a higher proportion of G's and C's, your TM is going to be at a higher temperature. If you have a lower concentration of G's and C's, it's actually going to be at a lower temperature. This turns out to be absolutely critical for things like PCR, hybridization, etc. A lot of what we do in molecular biology in the lab really has to do with this denaturation and renaturation. The other thing, which is not shown on here, but those base pairing interactions are incredibly specific. If you take two strands apart and then try and put them back together, they'll find the right base pairing interactions. And very rarely are they going to find the incorrect ones. And also, if you have a few bits that don't bind properly to each other, the base pairs aren't going to form. You're not going to form the nice double helical structure, which means you're not going to get that good stacking interaction. So this is going to be very unstable. So if you've got double-stranded DNA just by itself, even if the ends aren't very good, this is a very stable molecule. As soon as you start to have some mismatches in your DNA, this becomes very unstable. If the DNA can't pair with itself, it's very unstable. So it's just when you have these two complementary strands with each other that you get really nice DNA stability. And this, again, is a very useful aspect of DNA. So that's all I want to talk about in terms of the individual molecules. The structures, per se, I want to switch over a little bit and start talking about the bigger molecules and what DNA really looks like inside of cells. So sort of an outline of the end of today's lecture and a little bit of Monday's lecture. Start out talking about chromosomes and karyotypes that you get. This is just literally looking at individual chromosomes. Pretty amazing, although we're moving a little bit away from that now with whole genome sequencing. Human chromosomes, look at the human genome, a little bit about what's in the human genome. Shout out for the human genetics course, which I think is happening next term, in terms of looking at more details that are present in the human genome. How many base pairs are there in the haploid human genome? One of those numbers that Stedman expects you to remember. So 3.2-ish in the haploid genome, 6+-ish in terms of the diploid genome. That's as accurate as I expect you to be because people keep changing the numbers here, and it turns out from individual to individual there's more or less DNA in some of them. What's present in there, talk a little bit about the differences. So people always say you've got your mom's DNA and your dad's DNA. How different are they from each other? Turns out to be a relatively small amount. A little bit about genome evolution. Most important as far as this course is concerned is how you can go from a DNA, which actually here, remember I told you about the repeat structures. I should have emphasized those a little bit more. From Rosalind Franklin's X-ray diffraction of the fiber of DNA, I mentioned there were two major repeats. There was a 0.34 nanometer repeat. Turns out that's the distance between each of these base pairs. And there was also a 3.4 nanometer repeat. That's because the DNA wraps around every 10 base pairs. So every 10 base pairs, the structure is going to look exactly the same. So if you go from here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, it should be back on the other side. And so 10 base pairs per turn of your DNA in B-form DNA. And this will become also quite important as we move along here. But those numbers, 3.4 nanometers. 3.4 nanometers sounds pretty small, right? What about 3.4 times 3.2 billion? That starts to be a lot. A very, very, very large distance of your DNA. DNA is not terribly efficient in terms of encoding genetic information. As we talked a little bit before, there's actually not very much protein coding DNA in those 3.2 billion base pairs. So DNA has to be incredibly compacted inside of cells. But at the same time, that DNA needs to be accessible for replication, transcription, etc. So it's really pretty amazing how we can have DNA which is compacted yet accessible, and we'll spend a lot of time in the rest of the class talking about how some of that works. Of course, the first steps that we know the most about, nucleosomes, the wrapping around of the double-stranded DNA around histones. Then we'll talk a little bit about the 30 nanometer filament. The further and further away we get from this individual molecule, the less and less we understand about how chromosome compaction is going on. So let's talk about chromosomes. A lot of people say bacteria and archaea don't have chromosomes. This is completely wrong. They don't have nucleosomes that necessarily associate with their DNA, at least not in bacteria and not in some archaea, but still they've got exactly the same problem. Yes, E. coli has a lot smaller genome. It's only 4.5 million base pairs. But even 4.5 million base pairs, there's no way you can squish that into a single bacterial cell. So you have exactly the same problem. You have to compact your DNA, and you have to have it be accessible to all of the machinery that needs to interact with it. We'll talk about DNA repair a little bit later on. Turns out that DNA is a really crummy molecule for storing information because there's all kinds of chemical changes that can happen to it. So the interaction with proteins, which is what happens to DNA in order to get it compacted, is also important to protect that DNA from any kinds of changes that can happen to it. Any kind of chemical change that happens to your information, if that information is stored chemically, it's going to change the information. So protection is also a very important aspect of what we call chromatin. What's chromatin? Chromatin is just DNA plus proteins. Those proteins in some cases are going to be the histones, but there are other proteins that are involved in that process. The other aspect of these proteins that interact with the DNA is that they're important for allowing the DNA, once it's been replicated, to be pulled apart. And so if you think about cellular replication, you've got one cell goes to two cells, one genome goes to two genomes. Somehow those things need to be separated relative to each other. So it turns out that a lot of the DNA binding proteins, the chromatin proteins, are very important for this process of pulling these two things apart. So let's take a look at this process. An interphase chromosome, so think about the cell cycle. This is most of the cell cycle. This is where most of the activity is going on in any particular genome. Your DNA is being transcribed. That RNA is then going to be translated. Most of the time in interphase, if you just purify DNA, you'll see, oh, come on, let's go back. Change over to my pointer, that would help. This is what an interphase cell looks like if you just look at DNA. So there's a lot of DNA which is compacted here, which is very hard to define. There's still lots of proteins that are associated with this. Lots of people call this what's called the nuclear matrix, holding all of your DNA together. As soon as you move out from there, most of the DNA looks like this. And it's about 30 nanometers across with lots of white dots on it. Each of those white dots represent nucleosomes, and these nucleosomes are packaged together. A few parts of the DNA look like this, the so-called 10 nanometer fiber or beads on a string. These individual now black dots are the nucleosomes. So this is going from your normal cell, you break it open. This is 1,000 times compaction that you have. Most of the time inside the cell, the DNA is compacted about 1,000 times. However, for metaphase chromosomes, you have yet another 10-fold concentration to give you the chromosomes that you can actually see. And this is going right back to the last lecture. We talked about Friedrich Michner and also some of the studies that came out of Mendel's studies, seeing the individual chromosomes. You can see the chromosomes. That was what gave you the genetic information. So the only things you can actually see in the microscope, in a regular microscope, are these metaphase chromosomes. Again, this compaction is extremely well known, exactly how this works. This is a lot less well known, and even less well known is the compaction that happens here. However, because those metaphase chromosomes, you can see in the microscope, they're incredibly useful for looking at chromosomes, and particularly chromosomal abnormalities. So a normal human complement of chromosomes is shown here. This is in fact just the haploid version in a normal karyotype, which is literally looking at all the chromosomes in any particular organism. This is now metaphase chromosomes because they're nicely compacted. You've got 22 pairs of autosomes, i.e. the non-sex chromosomes, and then 2 sex chromosomes. This is true in humans, and this is the 4th edition, but it doesn't really matter here. Now, if you look at each of these chromosomes, you'll notice black bands and gray bands, etc. on them. That's due to staining. One of my ex-colleagues, Lester Newman, emeritus professor here at Portland State, he likes to call these barcodes. And so just literally looking at barcodes. And he looks at fruit flies, basically exactly the same thing. So any individual chromosome, doesn't matter from whom it is going to have this particular barcode to it. And people get really good at recognizing all of these barcodes. Another way of looking at chromosomes is now using that technique that we talked about, raising the temperature, lowering the temperature. DNA comes apart, single-stranded becomes double-stranded. If you put in little pieces of DNA that have appropriate colored markers, in this case fluorescent markers, you can get these karyotypes to now be different colors as opposed to just different bands that you see in black and white. So this is what's also called a rainbow stain, and this is the kind of thing that you would see if you'd done this kind of rainbow stain on metaphase chromosomes. So if you look here, anything that binds to chromosome 4, we'll just take chromosome 4 as an example, is going to be light blue. And that's because of the combination of sequences and fluorescent tags that are there. So you'll see there's one copy of chromosome 4 here. Here's the other copy of chromosome 4 over here. Here's one copy of chromosome 1. Here's another copy of chromosome 1. And so you can pull all of these things apart, line them all up with each other, and you've got 22 pairs of autosomes. And in this case, it's a genetically deficient organism because it's got these kinds of sex chromosomes in it. So that's the process of looking at these karyotypes. Why do people care about karyotypes? Well, one major reason is that by just looking at these barcodes, if you see a change, you see some kind of chromosomal abnormality, and literally just by looking at these barcodes and matching these things up with each other, here's just an example of one of these karyotypes. You'll see this is your normal case, and here we have an abnormal case because there's a piece of this barcode that's gotten moved over to here. This particular case is called the Philadelphia chromosome. Turns out that this happens in a particular kind of leukemia. That is why Brian Drucker at some point at OHSU will get the Nobel Prize. I will bet a lot of beer on that. Because he developed a drug which treats this chronic myelogenous leukemia due to this particular translocation. So if you can see this translocation in a karyotype, there's a drug that can be used to treat that cancer. Amazing. Just looking at a karyotype, you've got a drug that will actually work with it. It took a lot more work than just seeing that translocation and seeing what was going on there. But a lot of these things happen. So this is, I've been very fortunate again to be teaching this class for long enough that I'm like trying to take over the rest of the world, including OHSU. So some of my ex-students actually were working in the cytogenetics lab up there. And so they sent me these real karyotypes. So these are from the OHSU cytogenetics lab. These are normal human lymphocytes. These guys obviously know what they're doing, actually gal in this particular case, if I remember correctly, because they can take this mess and line them all up with each other and figure out where all of these pieces are. So this is your normal karyotype. This is great. These particular cells are fine. On the other hand, if you look at some other karyotypes, they end up being really, really bizarre. So this is a cancer karyotype. If we look here, Chromosome 1, there are actually 5 copies of chromosome 1 here, 3 copies of chromosome 2. It's completely lacking in chromosome 17. This is pretty typical kinds of cancer karyotypes you see, just completely messed up in terms of their chromosome structure. Yeah? The karyotypes that are trying to crawl off the page, the ones down here at the bottom? Oh, the top right here. And those just line them up. These are the ones they couldn't even assign. So this is really, again, amazing. Now, there's a lot of debate about whether it's this particular weird karyotype which leads to the cancer leads to the weird karyotype. That's still up in the air, and it probably varies from one cancer to another. This is another example, one of my more favorite ones here, and yet again, very strange kinds of things. Just one copy of chromosome 2, one copy of chromosome 3, one of 6, completely lacking 14. And just down here at the bottom, this is what I was asking you about. This is the karyotype that they start with. This is they start with this, and then they arrange all of them together here in order to give you your final structure. So karyotypes, I think it's amazing just to look at these things. These days, we're doing more and more DNA sequencing to look at these. So I'm hoping that karyotypes are not going the way of the unicorn or something else that are, I don't know, phone cords. There we go, phone cords. Which I've got a bunch over here as my helical examples as well. But yes, I think you can see a lot in just looking at these various different karyotypes. So what's the most important thing about looking at these karyotypes? Lining them all up with each other. You need to know how many to line up with each other. Everyone remember, or can go back and look at their notes, how many autosomes we have? Good. So, how many pairs of autosomes are there in a normal human karyotype? 1, 2, 22, 23, or 46? For those people who got here late, I'm going to be giving everyone at least 1 point today for just being here with their clickers. So you can check and make sure online that they're being registered. So look this afternoon to make sure that you have a clicker point. If you don't, send me your username and your clicker number. 5, 1, 0. What do we have? You're still messing with me. Try not to get 100%. So a normal human karyotype, if I'd ask this question about a cancer karyotype, what would you say? Absolutely no idea. That would have been the thing at the bottom. Could have been any of these. I think the maximum number of chromosomes that people have seen in some of these is over 50. 50 to 60 chromosomes. Just crazy stuff. And the autosomes are the non-sex chromosomes. So it's 22. Okay, finish up talking a little bit about the human chromosome. Again, this is something that we could spend a whole quarter talking about, actually probably even more than that. But just another quick description of sort of why we have to have all of this chromatin and condensation and so on and so forth. If you look at chromosome 22, you may or may not remember, in the karyotypes, that's the one that's just really, really tiny. It's actually one of the smallest human chromosomes, and in fact was the first human chromosome to be completely sequenced. Even then, if you had the chromosome here, it's only 50 million base pairs in length. And if you were just to go from 1 base pair to 1 millimeter, you stretch out the chromosome, it would go all the way across Africa. So I forget if that's just this chromosome or the whole human genome, but still a ridiculously long piece that, again, has to be compacted into a nucleus for most of the time. Still rather incredible. If you look at this particular chromosome. Turns out the human chromosome 22, as well as being pretty small, is actually pretty gene dense. It has lots of genes in it. If you look at just 10% of it, here we've got the genes as being these red and brown stripes. The white stripes are the junk DNA that we're not so sure about. 1% of that chromosome has about 4 genes in it. Again, this is a pretty gene-dense chromosome as far as the human genome is concerned. In that one gene, you have quite a few sequences which are known to be regulatory sequences, a few exon sequences. These are what's going to actually end up generating your final messenger RNA. We'll talk a lot about splicing later on. That messenger RNA will then be made into a protein, and that final folded protein is going to actually have your activity. The main thing to look at here is you see there's a bunch of gray, and the non-coding sequences, the junk DNA. Well, if you've got to do all this compaction, what the heck is all this extra DNA doing here? So, very open question. We don't know what's going on there. One thing I wanted to mention is that these little red stripes, the bits that are coding for protein, are usually pretty small. They're only between, in some cases, actually the smallest ones are just 8 nucleotides. Most of them are around about 150 nucleotides together that are coding for protein, individual protein sequence. And it's figuring out which pieces are protein coding and which pieces are non-protein coding, which is a pretty tough question to actually answer. But if you put all of this together, and this is one of the nice things about this textbook, 2015, we actually had a pretty good idea what was in the human genome. Again, about 3.2 billion base pairs, approximately 21,000 protein coding genes. This is almost an order of magnitude lower than what people thought before we did the first human genome sequence. People thought there were hundreds of thousands of genes. We know there are only about 20,000. That's actually not very different from worms, flies, etc. So that's really quite interesting. There are lots of variation in terms of numbers of exons, etc. But I wanted to point out a couple of things here which have changed even since 2015. This number of non-coding RNA genes, here they say approximately 9,000. That number has actually gone up to about 20,000 since then. We're discovering more and more of these non-coding RNA genes that are present in the genome. And then only about 1.5% of the genome is actually protein coding. So 98.5% of the genome is not protein coding or not coding unique individual genes. So why don't we just go ahead and leave it here. It's Friday afternoon. We've got a couple more minutes if people would like to come up and take a look at the DNA structure. I've got a bunch of helices over here, and I'll be hanging out having office hours here until about 11.